All right, this is what we did in class on Tuesday, January 5th. I just forgot to push record on Zoom. So this is just the basics of walking through a little modeling of meiosis. Most of this was covered in the meiosis lecture that was already posted, but this is just in case you're taking this class and you weren't in actual class on Tuesday, or if it just went too fast for you. This is what we did with the pop beads and how we filled in the chart. So I'm not going to stop and pause for you to write down notes. Just feel free to pause when you need to pause, okay? So to start off with, we're going to have 2 chromosomes, okay? And so here we are, 2 chromosomes, okay? We can tell they're different chromosomes because they're different lengths, okay? We have longer chromosomes, which would be one, and the shorter chromosomes would be the other. Any 2 chromosomes that are the same length are the same type of chromosome, okay? And so in this example, I have 2 of this type and 2 of this type, okay? So this is one type of chromosome. This is another type of chromosome. Why do I have 2 different colors? Well, one's from mom, one's from dad, okay? In somatic cells, this is human body cells, we have 2 copies of each kind of chromosome. So let's say this is chromosome 1 and chromosome 2. We have 2 copies of chromosome 1, one from mom, one from dad. We can tell they're the same because they're the same length. We have 2 copies of chromosome 2. Again, one from mom, one from dad. We can tell they're the same because they're the same length. All right? So this is what your normal cell starts out with. And as far as our vocabulary here, this would be diploid, because diploid means you have a copy from mom and you have a copy from dad. Okay, so this would be diploid. Our N here, remember N stands for how many different types of chromosomes are there. Our N is 2. And then our 2N is 4, because we have 1, 2, 3, 4 total chromosomes. We have 2 of one kind and 2 of another. So we have 2 different kinds of chromosomes. We have 2 shorts and 2 longs. Okay? So now we're going to move into interphase / prophase 1. In interphase, as we know, we learned about this in the cell cycle, a lot of things happen. We're just going to focus on what happens with DNA. And the main thing that happens with DNA in interphase is in the S phase, our chromosomes replicate. They duplicate, okay? So this chromosome from mom is going to make a copy of itself. And this chromosome from dad is going to make a copy of itself. They are now joined together. They are duplicated chromosomes. Okay, and this one from mom is going to make a copy, and this one from dad is going to make a copy, okay? And they're going to come together to form a tetrad. Tetra means 4, which basically means this copied one from mom and this copied one from dad, same type of chromosome, get stuck together in a little cluster. Tetra means 4, so they get stuck together in a cluster of 1, 2, 3, 4 chromosomes. Same thing over here. The copied, duplicated one from mom, duplicated one from dad, get all stuck together, okay? Otherwise, going on in the cell would be things like, obviously, typically we've got a nucleus here, but that nucleus is going to go away. We're going to have centrioles move to opposite sides of the cell. Okay, so if I want to draw in my cell here, I'm not going to draw a nucleus because it's just dissolved. I've got centrioles. This should be similar to mitosis. Remember that. Our spindles are going to start forming, but, you know, they're not going to fully form yet, my spindle fibers. And I got these tetrads here. And as it turns out, with my tetrads, in prophase 1, a very, very important thing happens, and it's called crossing over. Okay? And with crossing over, what happens is, in our tetrad, notice these 2 chromosomes touching. Well, as it turns out, they touch and squish so much that they actually end up swapping some DNA. How much? It's variable. But basically what happens is these 2 inside ones, like a little piece of mom's breaks off, and a little piece of dad's breaks off. Ooh, that one's a tricky one. Did not want to come off. There we go. And they swap. So now this little bit that was on dad is now on mom. Okay, and vice versa. This is called crossing over, and this occurs in prophase 1. Okay, so there we go. Still in a tetrad, okay, which is little bits exchange, and the same thing is going to happen with this tetrad. This happens with every single chromosome. And remember, in humans, we have 23 pairs of chromosomes. 23 times 2 is 46 total chromosomes, right? So this is happening with lots of chromosomes. I'm not going to model it. I'm just modeling this with 2 because obviously it would get out of hand quickly. Okay, so I'll put they wouldn't usually separate, but I can't do it without pulling apart. Okay, so there's my tetrads now. Crossing over has occurred. My cell right now is 2N, okay? It is duplicated, and it is diploid, okay? Because I have a copy from mom, a copy from dad. And then, just like in mitosis, in metaphase 1, the chromosomes are going to line up in the middle. So my spindle fibers are going to fully form, and my spindle fibers are going to push and pull these to the middle. It doesn't matter which one goes on top. This is just totally random. I'm just, okay, we'll go like that, okay? So we have our spindle fibers here, and they're the things that are moving the chromosomes to where they should be, okay? And there's going to be a bunch of fibers here. Still 2N, still diploid, still duplicated. Because notice my mom's chromosome still duplicated, so is my dad's. They're still together, same down here, okay? Anaphase 1, the cell's going to elongate, and the tetrads are going to get pulled apart. So they move to opposite sides. Our homologous duplicated chromosomes get pulled to opposite sides of the cell, like that. Okay, so these spindle fibers are going to shorten up, pull it to opposite sides. And then in telophase, again, still 2N, still duplicated, still diploid, because it's all in the same cell, right? And then we're going to go through telophase. We're going to get a nucleus reforming, going to get our spindles going away, all that good stuff. The membrane's going to pinch in. And then we're going to go into cytokinesis, where actually the cytoplasm splits, and we have two separate cells. So let's take a look at our two cells here. We'll put them in a nucleus. Get back in the nucleus. Okay, so here we go. All right, so I have two cells now. Let's take a look at diploid, haploid, duplicated, not duplicated. Is this chromosome still duplicated? Yeah, it is. I still got two of moms that are duplicated. So still duplicated. Is it diploid or haploid? Does it have a copy from mom and a copy from dad in each? No, this one has two moms, right? This one has two dads. So now we're down to haploid N, okay? I got just one long chromosome and just one short chromosome. They're duplicated, but in order to be diploid in 2N, I would need both of these in the same cell, okay? But they're in different cells. I just got duplications here, okay? So this is the end of cytokinesis 1, N duplicated haploid, okay? Then the cell is going to go, these two cells are going to go into a second division. No interphase, no S phase, no chromosome duplication. We're going straight into meiosis 2, and the whole point of meiosis 2 is to unduplicate these duplicated chromosomes, okay? So in prophase 1, again, my nucleus is going to go away. Okay, chromosomes condense into chromatin, all the stuff that we learned about in mitosis. Our spindles are going to start to form. Our centrioles are going to move to opposite sides of the cell. Going to get some spindles here. I'm still haploid. I'm still duplicated. I'm still N. Okay. Next, in metaphase, just like always, my chromosomes are going to line up in the middle. Which way? Either going to be like this or like this, whatever. It doesn't really matter. Just pick a way. Okay, and so again, the spindle fibers are what push and pull the chromosomes to the middle of the cell in the first place. And they attach here in metaphase. We are still N. We are still duplicated. We are still haploid. Okay, and then in anaphase, our spindle fibers are going to unduplicate our chromosomes. They're going to pull apart those sister chromatids. So this is going to get pulled over here, and this is going to get pulled over here, like that. Okay, and obviously these spindle fibers are going to shorten to do that. So again, this is anaphase 2. So now we are still haploid, we are still N, but no longer duplicated, because duplicated chromosomes are attached to each other. So as soon as anaphase 2 happens and pulls them apart, no longer duplicated. Okay? And now, lastly, we're going to go through full-on, telophase. Things are going to start to Our spindles go away, our centrioles go away. We're going to have 2 cells right over here with the nucleus. Boom, boom. This is now a cell with the nucleus. And same thing over here. Okay. These got separated, these got separated. La la. I'm going to erase this here. All right. So here we go. So at the beginning of meiosis, I had 1 cell. At the end of meiosis, I have 4. At the beginning of meiosis, it was diploid. I had 2 longs and 2 shorts, right? Copy from mom, a copy from dad of long, a copy from mom, a copy of dad of short. But now I just have 1 long and 1 short, so I'm haploid. Okay? As it turns out, I got a mom long and a mom with a little bit of dad short. Okay, that's the way that one turned out. But notice, are any 4 of these the same? No, none of them are the same. They all got different combos of chromosomes. So the end product of meiosis are 4 genetically unique cells, whereas the end product of mitosis are 2 genetically identical cells. Okay, and this is really, really important. Really important to have genetically unique cells. Otherwise, if that wasn't the case, you and your siblings, if you are a girl and you have a sister, you would be identical twins. But you're not. Most of you aren't, at least. Okay? We'll talk about that in a bit. So, this produces 4 identical cells. Are genetically non-identical cells. What I want to show you is what would happen if crossing over didn't occur. So I'm just going to zoom through this. You don't have to like copy this down, but I'm just going to zoom through it so that hopefully you see the importance of crossing over and leading to genetic diversity. Okay, so we're going to start in interphase. So wait, let's just start back at the beginning. Here we go. Diploid, N is 2, 2N is 4. Okay, chromosomes duplicate, form tetrads. Let's say crossing over does not occur. Okay, so then metaphase, they line up in the middle. Boop, boop. It doesn't matter which way. Okay, and this could be flippy floppy too. Could be like that. Okay, totally random chance. And then in metaphase, in anaphase 1, then our tetrads separate. Okay, and then by the end of cytokinesis, we would have 2 different cells. Okay, over here and over here. And then let's say meiosis 2 happens. Basically with meiosis 2, then we would go like this. We would go like this. Okay, and these would be our 4 resulting cells. Are these genetically different? No, these 2 match, and these 2 match. So you would have a lot more identical twins running around out there because we would have gametes that are identical, okay? So remember, this whole process is producing sperm or egg. But this is not what happens. Crossing over does occur, and because of that crossing over that occurs, we ended up with 4 genetically different gametes, which is why basically there's no one on Earth the same as you. Okay? Nobody on Earth the same as you. Except for identical twins. Okay? And so with identical twins, what happens, with identical twins, what happens is there's a sperm and an egg, and they get fertilized. And after fertilization, we have lots and lots of mitosis. So 1 cell becomes 2, they're identical. Becomes 4, becomes identical. 8, 16, 32, and so on. That keeps doubling and doubling and doubling. It all has the same DNA because it's going through mitosis. Okay? And at some point, within the first couple weeks of life, okay, kind of far into it, at some point, that blob of cells, that 64 cells, that 120 whatever cells, goes bloop into 2. And the 2 are genetically identical because it used to be part of the same blob, and it just went like this. And so both those 2 things grow into babies in mom at the same time, and they have the exact same, oh, there go my lights, the exact same DNA. That's identical twins. Okay? With non-identical twins, remember we learned that because meiosis produces 4 genetically unique cells, okay? So with non-identical twins, aka fraternal twins, what happens is you have the mom release 2 eggs in a month. Usually she just releases 1, but this time her body releases 2. So her body releases 2 eggs, genetically completely different, and if each one of those eggs, they will get fertilized by a sperm. This one gets fertilized by a sperm, and this one gets fertilized by a different sperm. And the sperm, again, genetically different. So we got one combo of genes here, one combo of genes here. They just happen to be cooking in mom's body at the same time. That's fraternal twins. And fraternal twins, always, 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 if you have a boy-girl twin, it's fraternal, right? Because boy-girl can't be identical, right? Okay. Sometimes girl-girl twins are fraternal, just 2 eggs that happened to end up being fertilized by X sperm. Okay, that leads to 2 baby girls growing in mom at the same time. Some boy-boy twins are fraternal, meaning 2 different eggs, 2 different sperms. They just grew in at the same time. But every once in a while, there's 1 egg, 1 sperm that grows, and bloops into 2, and those are twins, identical twins. Other than that, for the rest of us, with our siblings, we are not identical to them because the sperm and egg that made us are genetically different from the sperm and egg that made them. Okay? So that walks you through the process of meiosis, looking at some pop beads, seeing what it looks like with crossing over, and then understanding that if you take away crossing over, you lose that genetic variation. And I just want to let you know that it does, like I showed you what it looks like, crossing over doesn't happen, but crossing over does happen. On every single chromosome, between 1 and 3 times per chromosome, there's different crossing overs. So you get such a jumble that it's basically mathematically impossible to have one of your siblings look exactly like you unless you're an identical twin. Okay? So hopefully that helps.